Hi everyone, for chapter 10 and 11, we will discuss the process of turning DNA sequence into a functional protein. This happens in two main steps, transcription and translation. Transcription, which takes place in the nucleus, is going from DNA to mRNA, which we will discuss in this chapter 10. Translation, however, which takes place in the cytoplasm, goes from mRNA to an actual protein, and we will discuss this process in chapter 11. These are fairly simple chapters if you have a good background on these processes from your previous biology classes. Okay, so let's begin by talking about the structure of RNA. Just like DNA, RNA is a polymer of nucleotides joined together by, again, phosphodiester bonds. What differs is that RNA have a ribose sugar, okay, instead of deoxyribose sugar, meaning that at the 2' carbon position of the sugar molecule, and a free hydroxyl group is attached instead of just a hydrogen in DNA. Hydroxyl groups are more reactive than just a hydrogen, and hence this is what makes RNA less stable in a cell, which we have discussed in the last chapter. As we have already mentioned a few times in previous chapters, RNase has all the nucleotides, but substitutes thymine with uracil. Hence, uracil forms a complementary base pair with adenine. Okay, so you can see here where we were talking about that hydroxyl group bound to this 2' carbon of the ribose, and then here you have your uracil instead of the thymine. All right, so the third major difference is that RNA is only made up of a single strand versus double strand in DNA. Because it is a single strand, RNA can form other secondary structure, kind of like this, if there are any complementary regions present within its sequence. Contrary to DNA, there are many different classifications of RNA molecules. We will talk a bit about each of these in this lecture. The process of turning DNA into RNA for protein synthesis is called transcription. It is not exactly identical to the process of DNA replication. If anything, it seems to be a bit simpler, at least for your memory's case anyway, for the exam. For DNA replication, the entire genome needs to be replicated. But for transcription, we are only reading the gene that is used to make a needed protein product. Okay, not all of them are needed at the same time. We don't consistently need 100% of all our gene products all at the same time. So, genes are constantly being turned on and turned off in a cell based on needs. Transcription uses different raw materials, okay, enzymes, and only one DNA strand will serve as template strand. I'll talk about this in detail later in the lecture. All right, so scientists were able to prove that the process of transcription requires DNA as a template to make RNA product by looking at the electron microscopy, okay? What they saw was a Christmas tree type of structure, where there was a middle stem right here and shorter lines coming off of that middle stem. They were able to show that the middle stem was in fact a DNA molecule because when they add DNAs to the sample, the middle stem disappeared. And when they added RNAs to the sample, these shorter RNA molecules disappeared. So this provided a piece of visual evidence to support the process of transcription and that it involves both a DNA template to produce these RNA molecules. Remember, we mentioned that transcription takes place on only one strand of the DNA molecule. The one being read and transcribed is called the template strand. whereas the other unread strand is called the non-template strand. Again, this is something that is different from DNA replication. Okay, so which of the two strands will serve as template strand will differ between different genes. Sometimes it's read on one strand, like right here. Okay, will be read on this particular strand, whereas sometimes it will be read on the other strand, like gene A and gene C here. Okay, and it differs by different genes. Regardless which strand the gene is read from, just like replication, RNA products are always made in the 5' to 3' direction. Remember, I talked about how in DNA replication it's reading from 3 to 5' direction, which means the RNA that's being generated when it's complementary sequence to the gene is going to 5' to 3'. Okay? Adding one nucleotide at a time, okay, it will match the complementary base pair. So that is why the final RNA product is both complementary and anti-parallel to the original DNA template strand. Don't forget that a U is added instead of a T, and U is always paired with A here as well, okay? All right. So the gene that is being read involves what's called the transcription unit. The entire sequence of DNA needed for transcription to occur accurately, we need the entire transcription unit. There are 3 critical regions of a transcription unit. First, there is the promoter region. This is a DNA sequence that transcription factors and proteins that we're just going to collectively call it transcription apparatus because we don't need to know all the details of what binds to that region. But there is a bunch of stuff that binds at that promoter region, okay? So proteins will bind and initiate transcription, okay? Once this apparatus binds, without the promoter, transcription will not happen. It also tells us which strand of DNA will serve as a template strand, and hence which direction the transcription will proceed. For example, if the transcription was read this way, on this promoter, this particular promoter, then is actually going to be read going the other way around, and RNA will be synthesized going this way. Or if you're reading the promoter on this strand of the DNA, then you will read and synthesize RNA in this direction. So that's why depending on which strand that will be read as the template strand, it also determines the direction that transcription will happen. Make sure you remember that the promoter region is not transcribed. It is not part of the final protein sequence. The codons associated with those will not get added to the protein. It is merely just a location signal that tells you where transcription needs to start from. Okay, so the second region of a transcription unit is called the RNA coding region, which is right here within our transcription unit. This is just a sequence of DNA that actually gets copied into the RNA sequence. The third region is where the sequence tells you when the transcription needs to stop. Okay, so this is called the terminator region. Contrary to the promoter region, the sequence of the terminator region does get transcribed into the RNA molecule, and hence transcription stops only after it has transcribed the termination region. So right here, this red block pink bar here is where you have the termination region, and transcription does not stop until it's done transcribing that termination region. Whereas promoter, it never gets transcribed into the RNA transcript. That's why the RNA transcript does include that terminator region. Okay, so two terminologies you will often see when talking about sequences of genes is that those that come before the RNA coding regions, which we're talking about here, okay, this is usually called upstream, while those that come after it, which is here, okay, those are usually called downstream. So make sure you remember this, what upstream and downstream means, because when we put this in problem setting or discussion about genes, we always refer to these terms. The substrates being added are called ribonucleoside triphosphate. Okay, this is slightly different from DNTPs. Okay, we're not talking about replication. This is transcription. So it is RNTPs that's being added to the growing chain of RNA transcript. So the process of adding RNTPs is similar to replication in that they are added to the 3' hydroxyl group to form a phosphodiester bond, which is a covalent bond, and hence the direction of transcript is also from 5' to 3' as we have discussed. But for transcription's case, we do not need presence of a primer. It just starts where the promoter region was recognized. Okay, after the promoter region is where the first RNTP will be added. Okay, so the enzymes required for transcription to occur is also very simple compared to replication. There's only one main one you need to know, which is RNA polymerase. There's actually many other accessory proteins and variations of RNA polymerase that is used at different stages of the process, but we don't need to go in depth into them, okay? All right, again, similar to replication, there are 3 stages of transcription: initiation, elongation, and termination. 3 steps. During the initiation step, the transcription apparatus, remember that's those proteins and transcription factors I was talking about, they will recognize and bind to the promoter region of your DNA template. Even though each promoter region sequence for different genes is different, there are similar specific parts, okay? These are called consensus sequence, as shown by this example here with 4 different promoter regions. Okay, they still have parts that are similar. Of course, only the parts of each promoter region is shown here. There are many other sequences downstream of the consensus sequence and many other sequences upstream, but this is showing you where the consensus sequence lies between all 4 of these promoter regions. Okay, so the most common consensus sequence in bacteria are called the -10 consensus sequence and -35 consensus sequence. Both of them are essential for recognition of the promoter and allow your RNA polymerase to bind. If these are mutated, change, it can lead to inhibition of transcription of that gene. So now the next step that happened during initiation is forming a transcription bubble. Remember that in replication, we form a replication bubble, but now we form a transcription bubble instead. Now the unwinding of the double helix of DNA still needs to happen, but it is done by RNA polymerase as well, instead of having all those other enzymes we talked about in replication. The first NTPs is then added to the first nucleotide of the RNA coding regions. And then the rest of the transcription apparatus, which, like I said, were the protein factors, is then removed from the promoter region so we can advance to the next step of transcription. Then comes elongation, where DNA is threaded through an enzyme, RNA polymerase, that does all the unwinding like I was talking about, add new RNTPs, okay, and rewind the DNA template, okay, so it does it all. It doesn't need helicase and all that stuff, okay, and then again, this should proceed in the 5' to 3' direction as illustrated here, 5 to 3' direction as the molecule is being made. The movement of the enzyme from the promoter region to the protein coding region usually requires a conformational change in the enzyme itself, which is the RNA polymerase, but you don't need to know how exactly it changes, but there needs to be a conformational change in order for the RNA polymerase to move on from the promoter region to get to this elongation step. The replication bubble remains to be about 18 nucleotides in length. So for example, if this was the transcription bubble, then this is about 18 nucleotides long in length, okay? It proceeds down the DNA template, okay? And then RNA molecule is made. The last step of transcription process is called termination. And this is very simple. This is where RNA polymerase arrives at the terminator region, as we've discussed, okay, passed through it, has added the correct complementary RNTPs, and then it stops the process. The newly transcribed RNA molecule is then released from the enzyme, where it will then move on to the next process. Now, in the early days, after Watson and Crick has proposed the structure of DNA, they also proposed that genes and proteins are what's called collinear, okay, which says that the number of nucleotides in a gene is proportional to the number of amino acids. So a number of nucleotides is proportional to the number of amino acids, as illustrated by this diagram here. So this is your DNA template with your sequence coding for that particular gene you're looking at. It is transcribed into the mRNA molecule with the specific sequence. And this specific sequence will then drive the sequence of amino acid that's made in the final polypeptide chain of the protein. Okay, there is no cut, no trimming. It is directly proportional. However many nucleotides you have is however many sequence of amino acids you will have in your protein. That was the original hypothesis for the collinearity of the gene and protein. Now, this concept was not entirely wrong. It is true for most bacteria and viruses, but it was definitely incorrect for eukaryotic cells. For eukaryotic cells, we understand that only a very small portion of DNA sequence actually encodes a protein product. There are a lot of other DNA sequences that is not for encoding a gene product. Scientists also found that there are no large pieces of RNA molecules in the cytoplasm. So that's why we have two reasons here. There are far more DNA than required to encode the protein. We don't know what they're doing there, but yet they are there and they're not for encoding proteins. And hence, the collinearity between genes and proteins is not exactly correct. The large RNA because our genome is so big, if everything was transcribed, then we would see huge RNA molecules within the nucleus and the cytoplasm, but they find that they were absent in the cytoplasm. All right, so hence we conclude that RNA that is transcribed in the nucleus must have undergone some changes that shortens it up before moving into the cytoplasm. Again, visual evidence for this exists, okay, where these sequences are probably cut away before moving into the cytoplasm, and this is shown as evidence by the electron microscope image here. So what they did in this experiment here is they have a mixture of sample of DNA molecules, which is double-stranded, with its complementary RNA molecule, which are single-stranded. They first heat it up to separate these double-stranded DNA right here. So now they end up being separated in those places. Then they cool down the mixture, and where Of course, DNA will reanneal with its own complementary sequence, but now you have these RNA molecules that are also of the complementary sequence to the DNA. So some of them will reanneal with the RNA molecule as well. Now they found that there are long regions of DNA that ended up looping out of the RNA molecules, okay, when they were paired with them, of course, which is a very good indication that these looped out regions are the sequences that have been cut out. And on EM, electron microscopy image, you can see that there are stretches of DNA or RNA binding together, and then there's all these loops everywhere, loops. So they think those loops are parts of the RNA transcript that had to be cut out due to its size. And hence, with the data presented, we can conclude that RNA produced within the nucleus contains sequences that is not in the final mature RNA. So now we have to classify two different types of RNA in respect to transcription. We have pre-mRNA versus mature mRNA. M, of course, stands for messenger RNA because it is a message going from the nucleus to the cytoplasm. Now, pre-mRNA would contain protein coding regions called exons, okay, as we know it. And then there will be non-coding regions, which were those looped out regions we saw within the electron microscope image. Those are called introns. So, multiple exons can exist within a gene, okay? So here you have this image showing you two different genes. One is the ovalbumin gene, okay, and the other one is a cytochrome B gene. And you can see that there are these dark green rectangles here. Those represents your exons, whereas the lighter green bars represents your introns. Exons are your coding regions where it actually does become part of the final protein product, whereas introns, it is the non-coding regions that should be cut out. Okay, so the section that has been cut out from the mRNA are the introns, which again are common in eukaryotes only, but rarely exist in bacteria. So that's why that collinearity concept proposed by Crick was not entirely incorrect. They are correct in bacteria and viruses, but not correct for eukaryotic cell. There is no set size for introns. They can be large, they can be small. It differs from gene to gene. But they are usually larger than your exons. As you can see, these lighter green regions here are a lot bigger than the darker green regions here. Okay, so in order for a pre-mRNA to become a mature mRNA, we discovered that there must be multiple steps of what's called mRNA processing events. So these processing events has to be completed before your mature messenger RNA can exit out of the nucleus after transcription is completed. So it has to undergo these processing events. In a mature messenger RNA molecule, there are three regions that are important. You have your 5' untranslated region, or what we call 5' UTR, or the leader region. It allows for ribosomes to bind once your messenger RNA has reached the cytoplasm, so ribosome binding. And just like promoter, though, it does not encode any part of the protein. It's just part of the sequence of your messenger RNA. The second region is the actual protein coding region, which specifies which amino acid sequentially is added in the protein product. The nucleotides are usually read in sets of threes called the codons. So if this was your mRNA molecule, you have, let's say, UAG, GGA, etc. This is the 3 nucleotide, and you have your codon. The next 3 nucleotide, GGA, would then be the next codon. Okay, and each codon will be specific for bringing in one specific amino acid. The last region is called the 3' untranslated region, or the 3' UTR, sometimes called the trailer region, which plays a role in stabilizing your mRNA molecule and process of translation. Okay, some probably it has to do with some steps in translation that we cannot have or go without these 5' UTR or 3' UTR region, even though they're not coding the actual protein, but yet when we take them out, we know that they no longer can go through with the rest of transcription and translation. The next thing we will discuss are the 3 processing events that has to take place in order to turn your pre-mRNA molecule into a mature mRNA molecule. Since we were on the topic of introns and exons, and how introns has to be excised out of your final mRNA sequence, we will start with this process first. This process is called RNA splicing, which we're removing our introns away from the mRNA molecule and reconnecting all the exons together. So within the sequence of introns, there is a 5' splice site and a 3' splice site. Okay, so this is part of your exon 1, for example. This is your exon 2. Here is where you have your intron. So the 5' splice site and 3' splice site actually have pretty unique sequence. 5' splice site has to begin with GU, and the 3' splice site has to end with AG. So there has been research shown that changing these simple 2 nucleotide sequences will completely block RNA splicing from happening. So it's very critical you have these specific 5' and 3' splice sites within the intron. A third important sequence within the introns is called branch point, which is located about 18 to 40 nucleotides upstream of the 3' splice site. So here's that branch point, and from here to the 3' splice site usually is 18 to 40 nucleotide spacing in between. It is very simple, made up of just one nucleotide, which is adenine. And again, even though it's just one letter, changing it, either through deletion or mutation, again, will prevent splicing from occurring. So now let's talk about how these 3 sequences come into play for the process of RNA splicing. The protein complex involved in this process is called spliceosome, which is one of the largest protein complex in a cell. It is made up of a different type of RNA called small nuclear RNA, or snRNA, that forms complex with almost 300 other proteins. So the RNA splicing process actually happens in 2 steps. First, the pre-mRNA molecule is cut at the 5' splice sites. This separates your exon, for example, exon 1, from its neighboring intron. The 5' end of the intron will then loop back in this manner and bind to that branch point, okay, to form that lariat structure, just kind of like a T-loop, as we have talked about with the telomeres, but not exactly the same. But is that lariat structure, And this structure will then eventually be degraded further down the road and be completely gone, and our NTPs can be reused for the next set of transcription. The second step, so this is the first step. This is the second step of RNA splicing. So in the second step, we will cut the 3' splice site, and the 3' end of exon 1 will then be covalently joined with the 5' end of exon 2. Here is your 3' splice site. We're going to make a cut right there. And now you have free-floating exon 1 and the exon 2 further down the road. And then exon 1 will be reconnected with exon 2 to give you an mRNA molecule that is now only consisting of, for example, exon 1 and exon 2. An intron has already been cut out, and now it has just spliced out the non-coding region out of this mature RNA molecule. So if there are more introns and exons, which usually there are within a gene, this process will be repeated to remove more introns within the gene. So in addition to RNA splicing, 2 other processing events have to take place that adds modification to the 5' end and the 3' end of the mRNA molecule. At the 5' end, a 5' cap will be added. This is done by adding an extra modified nucleotide called 7-methylguanosine. So this is as shown right here. So along with a few more of these methyl groups that's added to the 2' hydroxyl groups on the ribose part of the nucleotide. So that's why this is part of that oxygen that was part of the hydroxyl group is now the hydrogen that's attached to it is now substituted with the methyl group, which is a CH3 molecule. So the function of the 5' cap has been shown to be critical for initiation of translation and maintain mRNA stability within the cytoplasm once it exits out of the nucleus. They also say that it plays a role in the removal of neutrons during RNA splicing. All right, so at the 3' end, a poly A tail is added. This is a chain of nucleotide adenine. Okay, it can be anywhere from 50 to 250 of them being added as the poly A tail. Interesting thing about this step is that there is no template involved. Previously, all that we have talked about with DNA replication, telomere replication, or even transcription usually requires some form of a template. But this step does not require any template and are just added to the 3' end on its own. So before adding the 3' poly A tail, though, the non-coding region must be cleaved. Okay, so this, for example, is your 3' end. Before we can add anything, there has to be some portions of that has to be cleaved because those are non-coding region. And this location is usually determined by the sequence or has a consensus sequence of AAU, AAA. And some pre-mRNA will actually have thousands of nucleotides chopped off from this end before able to add your poly A tail to the 3' end. Now this process of adding the poly A tail to the 3' end is called polyadenylation because you're adding adenine to multiple adenine to the 3' end of the mRNA transcript. So the function of poly A tail is to assist with the exports of mRNA into the cytoplasm, okay, facilitate attachment of ribosome once it has reached the cytoplasm for translation, and it also has a role in stabilizing your mRNA molecule. Now, the details of how it does this is either still unclear or something that we don't need to dive deep into. Okay, so when we combine all three of these mRNA processing events, we start with our original gene right here in our DNA sequence. After transcription, so transcription happens. So now you have your pre-mRNA molecule that is produced, and it contains both introns and exons, okay? Introns and exons, and it can of course have multiple of them depending on the gene. Then a 5' cap is added, and a segment of the 3' end will need to be removed or cleaved because it contains non-coding sequence that is unnecessary. All right, and then the last step is you have to add, not the last step, but next step is you have to add the 3' poly A tail at the end after cleaving those unnecessary non-coding region. And finally, you have RNA splicing events that has to occur to remove all these intron sections from the final mRNA molecule. And remember, those introns are removed in that lariat-like structure, which eventually will be degraded, and RNTPs will be recycled and reused for the next transcription. Now, the exons that remains after the removal of introns will connect together, form covalent bonds, and now you have a mature mRNA molecule that is functional and ready for translation. So let's take a break here so I can show you, or you can view the next video on how this whole process happens by animation.